David, we are live now, so you can start whenever you're ready. Please go ahead.
Okay, thank you very much, Ned. Well, please, again, members that are not from Yucatan team, please leave this room. We're gonna let you in 5 minutes before in the case of the Brazil team, and the other teams, well, they will be later. Now we're gonna start with the Yucatan team. Again, you have 20 minutes to have this nice and emotional conversation with the judges. So let's start right now. So here, I don't know if there's a judge that can want to give a step forward with the questions. Maria Jose, can you go first?
Awesome. So Yucatan, it's so nice to meet you. I was so excited to see what you were going to do and the simulations that we were going to prepare with C-Stain and Zippers together. And it's totally understandable that you couldn't do it because of time. But I'm just super interested in understanding what are your expectations from that simulation and how different do you think that it's going to be from the thing that you had individually?
So if I may answer that question, well, we had a lot of expectation in those simulations because, of course, to be honest, here in this team, we don't have that much experience with simulations, but at the same time, it was very innovative, like new, and we wanted to see how these things were going to work. Originally, it's not known how the intermediate filaments interact by natural means, like inside the cell. So we wanted to have a hint in order to see if we can improve. And for that, we started to see, okay, let's simulate something with a part that we can control and there's a lot of information about. And then that's how we thought, okay, the leucine zippers are like the most known dimerized interaction we have. So we wanted to see, can we really optimize the way these filaments dimerize together and see a little bit of more information about how they interact. So the expectations we had is to see and learn, to have new information to work with and see if we can even improve the formation. Of course, this is only a simulation. There's a lot of matters that we have to consider after this part of the process. But if we can find a stable structure and an interest interaction between the leucine zippers in order to assure that this dimerization is going to occur inside another cell that's not the original, that's not the GTC, the glands on the hagfish. Like if it happens in E. coli, that's going to be a great improvement because we are going to reduce the purification processes and that's going to help a lot. So we know that we had to do a lot of simulations in order to see how are we going to do it, since it's a system that is not really very well known, the interaction of these domains. But we decided, yeah, let's do it. Because if we don't do it, who is going to? And that's our motivation.
Thank you.
Maybe next judge, let's give a round with the judges, and then if we have time, we start again. So if there's another judge that wants to give, okay, we're going to Mariana, and then we Nathan.
Hello, guys. Well, first of all, I was thrilled to read about your project. And I see that your main background was about microplastics and how they are accumulating in the Yucatan Peninsula. So I liked your approach on how you try to generate a solution to avoid the generation of these microplastics. But I was wondering whether at some point you thought of maybe proposing something that would degrade or decompose the microplastics that are already accumulated in the Yucatan Peninsula.
Yeah, the microplastic is a issue in Yucatan and or Mexico. So that the idea or working in a substitute or microplastic or fiber textile is our the what I understood the idea, but no, Agustin.
Yes,
Thank you.
of course. Yeah, let me continue. Don't worry, don't worry. First of all, there's a member of our team that is trying to access. Can you please give it access? Now, I will answer that question. So, honestly, we tried first to approach the problem that in a way that we don't allow it to happen. We are not really interested in like biodegradation. We know it's a very interesting topic. We know that, and that we could very well treat the problem in that way because there's so much room to improve in the water of Yucatan. This place is, well, very special in a lot of manners. And first, there's almost no dirt. And then second of all the micropollutants very easily get into the water here. And there's not a really very well treatment of those waters. So we know that that problematic it could be treated in other ways, but it's not like related that directly to us, to our project, because we kind of approach it in a way that we intend to do this textile product that is not going to have that issue in the future because it's a biodegradable product. We kind of approach that way more than directly solving, or I mean, degrading what's already there. We are like, okay, with this, if we start to produce textiles that are this way, the contamination is going to get, well, it's going to be less, a lot less, and we are going to help to solve that problematic in a way that we are not increasing it. So your question is very interesting. Like, we are trying to solve the same problem, but maybe in another perspective. Maybe we are hitting it in another point. We could, of course, treat the water, and maybe the project could have gone to another completely route. But in the end, we decided to approach with this. We solve the problematic, preventing it to happen, like that way, preventing it to increase. And that's how at least I think my team sees it.
Okay, great, guys. I understand that you didn't try to biodegrade these microplastics, and I really like your approach, and I think it can have a broader impact on how these sustainable development goals can be reached. So congratulations, and yeah, great project.
Okay, thank you, Mariana. And then we go with Alan.
Yes. So thank you, guys. I really enjoyed, again, I echo what the judges say. It was really nice to see your project, especially this connection between the textile industry, which is so important economically for Yucatan, and the unique environmental issues of microplastic in the special soil or lack of soil in the peninsula. I have a question about your device design. So I see you're using a constitutive promoter, a strong RBS, and a high copy plasmid. Do you anticipate any difficulties or adverse effects on the cells from such a high level of protein expression?
Okay. We don't really think about it because the other teams of the iGEM, they can use it, so they can test it. we have a little difficult in this project. It can be practiced and can really test it. So we see the other teams, they have the practice and they are produce the filament intermediaries, and the height, the mount, it's functional. So I think that it can work. So I try it, and if we practice in all these systems, maybe I can find the problems, but this moment I can just see. Only we can design and hope the better.
Kind of complementing my comrade here, we at least the work that was done because of the HQST 2020 team, they were kind of the predecessors of this project. So they did an excellent job looking what the results were, and they kind of give us a sense of, okay, this will work. And to the 2014 did exactly kind of very similar genetic circuits, because we're kind of focuses in the simulations mostly. So we were like, let's not complicate with something that's very like we don't really know. We are already going to something that we don't know, because we are adding this leucine zippers and cysteine. We could, of course, add more into the purification and methodology, but we decide not to, because we already have kind of uncertainties inserting that part. So if we, in the end, start adding only new things and new things and new things, well, then what's the object of study? What are we going to focus? There's a lot of information that we're going to have, and we have to focus only one. So we are like sure in the sense that we know how it's going to go, because already other team did this part. But at the same time, of course, we have to consider what the modifications are going to do, what the how to say it, how is it going to handle being in a real bacteria in vitro. Of course, there are some things that we may don't know, or if the expression is going to be as we want, but we intend to improve for.
Aí eu adoro esse macacão.
In this experience.
Você amarra de frente e de costas? De frente, na lateralzinha.
É o seu?
Uhum.
I'm sorry, please, members that are not Yucatán, please be on mute. Please, members, just members of Yucatán are allowed to speak to the judges. So be here, please put yourself on mute. Now, go ahead.
My comrade Peña is going to talk. Hold on.
Yeah, I just wanted to compliment and I guess summarize the information here that my peers were talking about. And essentially what they're trying to refer to is that the research itself has to be a very specific question that you're trying to answer, instead of trying to answer multiple questions given the limited amount of time that we have with this project. And so our focus was not so much factors such as biocompatibility with the cells. We're gonna have too much hard of a time doing that. Our main objective with this research and with the question that we're trying to answer is, can we build a mathematical model to make sure that in the future, those questions can be further answered through other different studies, whether in vivo or in vitro. And that is essentially what our team focused on during this project, basically allowing now to build on that information and be able to answer questions like that.
Great, thank you. I think, yeah, I think the three of you said it very well. This is more to bring attention to a potential issue that could come down the line once you start implementing this biologically. So thank you again.
Thank you, Alan. Now, Emiliano, you go.
Hi. First, congratulations. And I really love that project because I'm working with dyes. We do a lot of prototyping in my company with fibers. And so you are welcome to send samples when you have something. I wanted to know if you imagine how it will perform with natural or synthetic dyes, the filament.
Can you repeat your question, please?
Yes, if you have some idea of how it will perform when mixing with the natural or synthetic dye.
Okay, I have the response for this. Okay. Yes, we don't know. Thanks, because actually, like the project before this was the most recent one was the 2020 of the HKU's team, and that was the focus of their project. Like, can we add dyes to these filaments? They of course didn't. Their system of purification is different from us because we are synthesizing this inside of the cell, but they produce the subunits and then they add the dyes like with copolymerization and other things. But that was the focus of their project. Like they intended to add these natural dyes, and it was possible, and they were successful on doing so. So yeah, answering that shortly, yes, it's possible. And we really didn't do it because, well, that's not the objective of our project, but we knew that it was possible in the N terminal and C terminal points of the chains. You can add those dyes, or even you can add one color in one part and other color, and they combine. You can do a lot more colors that way. It's very interesting that work of the other team that was their main focus, and it was very interesting. Like, yes, it's possible.
Thank you.
Okay, thank you. Now let's go with William, please.
Thank you very much for the project you came up with and for how you presented it in your the Joggle website. My question is very simple. Have you or will you perform any mechanical or biological characterization of the native biopolymer from the original source?
Can you repeat the question, please?
So, okay. I just wanted to find out whether you'll perform any mechanical or biological characterization of the native biopolymer from the original source.
Yeah, well, focusing on the filament intermediate, the mechanical test is I don't we don't in the methodology is the last the last stage, but in the biological test in the cell well, not no more tests because only focus it in the church on the genetical in the church on the genetical sequences because it's a product, it's a bioproduct in the intermediate filaments, but in a mechanical test in the intermediate filaments, I no how do you say?
It's fine. I think I can compliment you, Freddie. And I guess what my partner here is trying to say, one of the main factors that comes into play when we're trying to, do all these things that we would, want to do, such as mechanical testing, is funding. And we would need to write or find the right resources to make sure that we have the, the correct funds and the proper equipment in order to do something like that. Now, if the project was, going to continue further, let's say we find, I don't know, a startup company or some other institution organization that is, interested in our project and willing to, let us borrow some of their resources. then we could potentially start looking into something like that. I was looking, for example, in the team from 2014, the Husk, and what they found out is actually that their predictions in terms of like the, I don't know, modulus of elasticity and all these properties actually matched up pretty well because they had actually gone into some mechanical analysis themselves. So it was not the most, I guess, rigorous analysis, but to a certain extent, they were able to, you know, back check that information that they had predicted. And so as for us, we don't have that luxury just because, you know, resources become the main issue here.
That sounds good. Thank you.
Thank you very much, William. We're going with Hector right now, please. We're almost on time, so please try to be short with your answers.
Okay, I'm terrible with my English, so I'm gonna read my questions. Yucatán, first of all, I would like to congratulate you for the great work. I only have a couple of questions. First, why you use a constitutive promoter to express your proteins, and is bacteria ideal organisms to produce proteins from higher organisms, and why didn't you choose a yeast?
Sorry, what was the, like, we heard like the end of the question, but what was the first part?
Okay, the first one is why you use a constitutive promoter to express your proteins.
Okay. Well, we kind of use it because in the end, it was already shown that that promoter was very effective in producing exactly this, at least this filament. It was already being like tested. So we kind of went there like, okay, to be sure that this is going to work, we're going to follow what the theory says and what the other team have done. And we can say, okay, this is reliable because the other team kind of went into a comparison of different promoters. So they kind of did that part, and we saw that and kind of analyzed the results and see, okay, this produces what we want almost surely. Actually, now that you're talking about it, like going shortly into maybe other matters, we were kind of worried because of the cysteine zipper about the post-traductional modifications that we could have to need, because cysteine zippers work that way. But and that point, we kind of consider, okay, maybe we want to do it in yeast. But in the end, we wanted to be more efficient, and bacteria is more efficient than yeast in that sense. So we kind of went with what we have to. We saw the information and we decided, okay, this is what we're going to do. We decided it that way.
Okay, thank you.
And I guess just to briefly talk on that, I know we're short on time, but like my friend said, it's a lot more efficient to try and incorporate some new genetic material into bacteria than it is for a yeast in that regard.
Okay, thank you very much. Now let's go with Felix. Please try to keep it short because we are almost on time, almost over time.
Ok, hola. Hola, bueno, antes que nada me disculpo un poco, bueno, como comentaba a David, bueno, tengo unos problemas por ahí, entonces estoy algo medicado, entonces voy a hablar un poco en español porque no creo que mi cerebro carbure en dos idiomas, ¿no? Con trabajo y carbura en uno a veces, entonces voy a tratar de hablarles en español. Entonces, bueno, antes que nada me encanta su proyecto, creo que es bastante interesante. Bueno, yo estoy más enfocado a los temas comerciales, entonces mi pregunta va dirigido a eso. Lo que noté es que no han trabajado tanto esa parte de, creo que solo es como una prospección hasta el momento, pero ustedes, ¿qué consideran o cómo consideran que pueda ser utilizado su producto ya? O sea, ¿quién podría ser su comprador? porque creo que lo tienen como que ustedes pueden darlo a otras empresas o como proveedores para que también ellos generen modelos de emprendimiento. Entonces, ¿cuál sería realmente ese modelo económico? Porque no me quedó muy claro.
Bueno,
Try to make it in English, please.
Okay, for the moment, it's only research, as you say, but we communicate with a lot industrial empresas, so they can pay some of the they can pay for the research, and they can take it the intermediate filaments to implement in your clothes or making your textiles. So the model is to make search and invest in research and make it, they can use it for your own textiles. So that's the idea generally.
Sorry. Like, yeah, because we're short on time, I'm going to like kind of room zoom this, but we have done like a very simple merge study in the way that like, okay, what is going to be our objective? We, just as Anai said, like we are going to be, for now, like, okay, we are kind of a raw material, not raw material, that's not the way that you say it, but like, yeah, we sell this idea of this implementation of clothes to another, for example, bigger company or something like that. And then that's where, okay, we can bring you this system that produces textiles in a biodegradable, sorry. And well, they can be produced this way, and it can give you this. What do you think about that? And well, that's how we at least plan to have. To be honest, like we haven't really go deep into that matter. You are right in that sense, but we, of course, as the project going to progress and we continue, we are going to see that more closely. For now, we are only looking it for that way because that's the more possible and easiest way, to be honest, to apply the project. But maybe as we get closer to a real product, we may start, well, we already start, but we may focus even more about what we want to do with this product.
Well, thank you very much. We are very, very short on time, so we appreciate very much the presentation of this very first class of Yucatán team here in iGEM Design. Congratulations, at least in room 5, you are the very first one. So congratulations, hope to see you next year in Paris. And please, let's go with Brazil team. Thank you very much.
Thank you too.
Thanks a lot.
Thank you for looking at the project, and I hope that you will like.
Now, please, I'm gonna ask very gently to all members of Yucatán team to leave the session. So we go now with the Brazil team. So try to keep it very fast to get together. And to the judges, please, let's make just one single question per judge. If we have time, we make a second round, but we are very, very short. We have many judges, and we have a very, very good teams that want to explain as much as they can all the very hard job that they have done through the year.
Goodbye, everyone.
Thank you very much. And advice to all other teams, please, if one of your mates answer a question, try to do not repeat the same. I mean, if someone already answered correctly, if you want, you can add something, but try to not humble around the same ideas, right? And let me remind you, please, to all members of the team, rename yourselves as the name of the team and your personal name, right? So now all Yucatán members, please leave the session, and we must have only Brazil members ready, okay?
Hello, everyone.
Hi, how are you?
Fine, and you?
Great, thanks.
We want to, first of all, we want to know if you have a translator. I don't know if you have it.
Yes.
Okay,
I am here to act as a translator for CNPEM team. Eu sou o tradutor aqui hoje.
Thank you.
Awesome. I didn't know that, but great.
Então, primeiro nosso time quer agradecer a presença de todo o júri e também agradecer pela tradução que a Steph proporcionou para a gente.
So they're thanking the presence of all the judges and also my presence here.
Thank you very much.
So let's start with okay. So let's start with this meeting. I don't know if there's any judge that wants to give one step forward. Any judge?
I can do it again.
Okay, great. Thank you, Maria Jose.
Awesome. So nice to meet you, Brazil. I have a question with respect to the proteins that you are going to put in the membrane for getting the light, right? So what I know is that you're using a BioBreak from China 2016, and in that project they were using the OMPA protein linked to the PBR, right? So I'm wondering, how are you linking the OMPA to the two proteins that you have, and also how was that design thinking to decide where were you going to put those proteins, in the N or the C terminal?
Maria, I'm going to have to translate the question to them, I think. So I am not familiar with their project, so I might need to take a little back and forth with you. So let me just ask them how exactly they want to go with that. Pessoal, como vocês querem fazer? Vocês querem tentar responder por vocês mesmos? Vocês querem que eu traduza tudo o que eles disserem, aí eu traduzo o que vocês disserem? Como que vai funcionar essa dinâmica?
Seria legal se pudesse traduzir tudo, porque tem pessoas do nosso time que querem também participar e eles não falam inglês.
Tá bom. So I'm going to translate basically everything, okay? So I'm going to translate back and forth everything that's going to be said. Pessoal, a pergunta deles é a seguinte: vocês usaram um BioBreak da equipe da China de 2016, e naquele projeto eles utilizaram a OMPA para poder localizar a PBRR na superfície da E. coli. As proteínas de vocês são ligadas ao OMPA? Ela colocou um GPR aqui. Como foi o processo de design, né, para decidir como que seria feita essa ligação da OMPA, se no C terminal ou no N terminal da proteína?
Para a construção desse BioBreak, a gente usou essa parte composta do time da China, que era uma parte que já tinha sido caracterizada. Elas têm um link entre os dois BioBreaks, né, dessa composite part, e a gente utilizou devido a que eles tiveram sucesso na utilização disso para a mesma finalidade que a gente. Então, a gente encontrou no registry uma parte que tinha a mesma finalidade e a gente alterou ela para auxiliar no nosso projeto, do nosso desenvolvimento.
Okay, so they chose this part because China already used it with success. So they just altered the composite part so that it would fit their project. Eu acho que tinha uma parte da pergunta que se referia a essa escolha de fazer o N ou C terminal. Tem algum insight específico sobre isso para adicionar?
Em relação a isso do N e do C terminal, a gente basicamente já seguiu de acordo com o design que já tinha sido proposto anteriormente. Então, a gente construiu a nossa parte em cima daquilo que já tinha sido feito. Então, a gente não participou dessa parte do design. A gente utilizou uma parte já caracterizada.
Okay, so they just they didn't give much thought about where to select regarding N or C terminal. They just followed whatever design was already made with the composite part that already existed.
Okay, thank you very much. Okay, next go with Alan, please.
Okay, thank you. Thank you, team, for sharing your project with us. I was really happy and excited to read it. Carlos, I will type my question also in the chat, but I will say it first.
Thank you.
I see that you use the genome sequence of E. coli to make a genome scale metabolic model. I work with these a lot, so I thought that was really cool. But I wanted to ask, why did you go through the process from the beginning of going from the genome, gap filling, and generating your model, as opposed to using a published curated model? And related to this, you had some predicted results of the growth of E. coli. I want to clarify just what were the units of measurement that you reported on figure 5?
Thank you. Pessoal, ele achou legal vocês terem feito um modelo metabólico, né, a partir do genoma, é algo que ele faz bastante. Então, ele está perguntando sobre isso, né? Vocês usaram o genoma para construir o modelo em escala genômica dessa bactéria, o que ele achou muito legal, mas ele queria saber por que vocês passaram por todo o processo de fazer a construção desse modelo, ao invés de simplesmente utilizar um modelo que já havia sido publicado e previamente curado. E ele tem uma segunda pergunta, que é para clarificar um pouco com relação aos resultados. Ele quer saber qual é a unidade do modelo de predição de crescimento da E. coli na figura 5 do trabalho de vocês.
A gente resolveu fazer desde o começo, porque a ideia inicial era que a gente pudesse escolher um chassi que fosse suficiente para o nosso projeto. Então, a gente queria, na verdade, utilizar uma outra E. coli que não estava nos bancos de dados futuramente. Então, seria uma E. coli que ela já é resistente ao nosso bioproduto. Então, foi por isso que a gente começou a aprender desde o início. E também foi um processo de aprendizagem, porque a gente queria aprender a usar os softwares das ferramentas computacionais para entender como o modelo era construído, até chegar nessa parte, mas infelizmente a gente parou uma etapa antes, porque a gente não conseguiu adicionar as vias que a gente gostaria e editar o modelo da forma como a gente queria.
Deixa eu dar essa resposta primeiro e depois vocês comentam sobre a outra. So they decided to go through all the steps because they wanted to learn how to create a genome scale model, especially because in the future they want to create a model for an E. coli that does not already has a model created for it and curated. So they wanted to learn how to do it so they can do it again in the future with another strain. As for the second part, I'm going to ask them to answer now.
Eu queria entender se é a figura 5 que ele está dizendo da curva de crescimento.
So your question is about the growth curve, right?
Sim.
Yes.
Sobre qual é a unidade? É isso?
Qual é a unidade de crescimento?
Aqui a gente está em número de células na growth curve. Começando de duas, a partir de 2 células.
So the unit is number of cells.
Okay. Yeah, thank you very much.
Thank you.
Thank you, Alan. Next judge, please.
Thank you for your project. I really enjoyed reading it. I just have one question. How does the light intensity affect the production of your 1/8 cineole? And is it a linear or generally, how does it affect your production?
And can you paste it on the chat?
Oh, sorry. Let me type it. I never typed it out.
But I got most of it. And the question is how does the light affect the product, right? The production of the product or something like that, right? Ok, então a pergunta é como que a luz afeta a produção do produto de vocês?
João, você pode responder?
Yes. A produção de um 8 cineol, no caso, ela passa por uma via metabólica do mevalonato, e antes de chegar na via metabólica do mevalonato, a gente utilizou uma via chamada Hef-Udd-Lundahl formulase. Essa via utiliza NADH e também ATP como cofatores. Pode traduzir essa parte, eu falo o resto, não vai ficar meio longo.
Okay, so I'm going to translate in parts. So the first part of his answer is they're using a pathway that uses both NADH and what else? O que mais que ele usa? NADH e?
ATP.
Yeah, and ATP. And acho que agora você pode seguir a resposta.
Ok. A regeneração de NAD quando você pega e insere um quantum dot associado à sua bactéria, você aumenta a quantidade de elétrons disponíveis dentro do seu sistema. E esses elétrons podem ser carreados por algumas partículas, no caso, por algumas moléculas, quero dizer, que são moléculas redutoras, e isso pode, de alguma forma, aumentar a concentração de NADH, e dessa forma você empurra o equilíbrio da sua reação para o outro lado.
Okay, I'm going to try my best to translate that, but from what I can get, when they use a quantum dot on their project, that'll help keep the concentration of NAD8 higher, and because of electron carrier proteins, that'll help to push the equilibrium towards producing what they want.
And if they increase the light intensity, does it affect how much they get?
Se você aumentar a intensidade da luz, aumenta o quanto você consegue aumentar a produção?
Sim, a intensidade da luz vai aumentar a quantidade de fótons que estão vindo para a nossa nanopartícula. E a quantidade de fótons que chegam nessa nanopartícula é a quantidade, de certa forma, proporcional à quantidade de elétrons livres na superfície da nanopartícula. E essa nanopartícula vai liberar esses elétrons e isso vai ser carreado. Então, a intensidade da luz afeta, sim, a nossa produção.
Yeah, okay, so the light intensity will increase the amount of photons that are reaching the system, and that will increase the amount of electrons that are available at the surface of the material, and that will increase its the production of the product.
Thank you.
Thank you very much, William. Next judge, please. Héctor.
Okay. Hello, Brazil. Congratulations for your work. Sometimes we make the mistake of believing that the modification or insertion of a gene will affect only one metabolic pathway. But in reality, we know that it can be a cascade of modifications. The expression of one gene can activate or suppress the expression of another, and that in turn affects yet another gene. So how do you consider this effect in your computational models?
Okay, do you have it on text so you can paste it, or maybe just?
The question is in the message area.
Okay, but is it already there?
No, I send it to
more.
Okay.
Okay. Thank you. Ok, então, às vezes, a pergunta dele é, às vezes a gente acredita que quando a gente faz uma modificação de inserção de um gene, isso vai afetar só uma via metabólica. Mas, na verdade, isso pode gerar toda uma cascata de modificações. Então, a expressão de um gene pode ativar ou suprimir a expressão de outros, e isso, por sua vez, pode, enfim, criar toda, né, uma bagunça ali. Então, como vocês consideraram esse efeito, né, em cascata de modificações no modelo computacional de vocês?
Atualmente no nosso modelo, né, a gente se baseou em modelos que já eram da literatura, que propiciavam esse tipo de estruturação dessa modelagem, né? O João ele pode explicar melhor até. Então a gente se baseou nesse tipo de modelos que eles já estavam sendo desenvolvidos e que eles contemplavam esse tipo de design, né? Mas se o João quiser complementar.
Vou complementar. Bom, no nosso caso, nós tínhamos duas perguntas que nós gostaríamos de responder com a modelagem matemática. A primeira pergunta era quais enzimas eram mais relevantes ou que tinham uma influência maior na produção de cineol e na redução de alguns intermediários. E a segunda pergunta era como o quantum dot ia afetar a nossa produção de 1,8-cineol. Pode traduzir essa parte.
Acho que talvez eu vou pedir vocês para tentar ir jogando talvez alguns bullet points, talvez escrevendo a resposta de vocês no chat, porque às vezes elas ficam muito complexas e eu dificilmente vou conseguir traduzir mantendo toda a riqueza de informações que vocês estão passando. Mas eu vou te pedir para repetir, inclusive, já que eu consigo passar um pouco mais claro.
Ok. Bom, a nossa modelagem teve dois objetivos centrais. O primeiro era entender como a luz poderia influenciar a produção de 1,8-cineol.
Okay, deixa eu com essa parte. So their modeling had two main objectives: to understand how light would interfere in the production of the product.
E também quais enzimas eram mais importantes durante o pathway, a rede metabólica.
And also to understand which enzymes were the most important for their pathway.
Deste modo, nós não trabalhamos de fato com as concentrações das enzimas em cada passo, e sim trabalhamos com a concentração relativa das enzimas para ver qual delas tem mais importância e não exatamente quanto que a gente precisava de cada uma delas.
So they focused on working on the relative concentration of the enzymes, so that they would try to find which ones were the most relevant for their systems. We have a question here from their mentor that she's asking if she can help with translation. I don't know. I'm going to pass that question for the moderators.
No, I'm sorry, but mentors are not allowed to help in any way to the teams. I'm sorry.
Ok.
Deste modo, o nosso modelo ele não prevê todas as reações que estão fora do pathway, por causa que o objetivo principal da nossa modelagem não era prever isso, e sim entender quais enzimas eram mais importantes.
Okay, so their model does not address that question about how other reactions on the system might change, because it was focused on understanding the priority of relevance of enzymes on the system. So they were trying to find which enzymes were the most important.
Great, thank you.
Thank you very much. And we still have time for one. Bueno, Emiliano, yeah, you go.
Okay, only one question. Have you thought of how much space you will need to make a bioreactor plant to eliminate, let's say, 1 ton of CO2, or do you have a number related to that?
If you can paste it, but I will start trying to translate it for them. Ele está perguntando se vocês pensaram com relação ao tamanho da planta industrial que vocês vão ter que ter, né, do tamanho do bioreator que vocês vão precisar para poder eliminar uma determinada unidade, né, por exemplo, 1 tonelada de CO2 ambiental.
Nesse primeiro momento, a gente não estava trabalhando com os valores numéricos de quanto a nossa bactéria consome, né? A gente buscou a gente fez modelagens qualitativas, e com esse reator a gente buscou fazer um hardware que propiciasse o nosso trabalho em bancada de forma segura para o time. Então, o objetivo do nosso hardware no momento era facilitar a consecução na parte de building. Mas a gente imagina que para subir para uma escala maior, a gente precisaria também desenvolver um processo maior também. Aquele reator, no caso, ele era de escala de bancada.
Okay. So they didn't focus on an industrial scale right now with their system. They were mostly focused on a bench scale for their model, and that's what the objective was, to assist with the design of the hardware for this part, for this moment of this project. So in the future they might do it in another scale, right?
E a gente imagina que com esse reator a gente pudesse poder coletar mais dados experimentais para construir modelos metabólicos mais precisos para chegar nesse ponto.
So with their current hardware, they imagine they will be able to collect experimental data to improve their system, right, their model.
Okay, I understand. Thank you.
Okay, thank you very much. We still have time for one single question, so keep it short, please. I don't know, Mariana or Felix?
There are no questions for me.
Okay, Felix?
Tampoco, ninguna pregunta.
Okay, thank you very much. Well, it was a real big pleasure to have a Brazil team. Thank you for presentation. It was a great job. And now let's go with the team from the Tech of Mexico. Thank you very much.
Ok, thank you all. É isso, pessoal. Obrigado para todo mundo.
Obrigado a todos.
Thank you to the translator, first time in iGEM.
Thank you, Carlos.
You very much.
Okay, bye-bye, people. Have a nice
first time
event.
in iGEM in general, design or general iGEM. So thank you very much for your help.
No problem. Anytime. Bye-bye.
Bye.
Bye, thank you.
Bye, thank you. Thank you to all Brazil members. I'm going to ask you gently to leave the session, and let's start with the team from the tech. Members of the MikuyTec, are you ready?
Yeah, just wanted to clarify, we are from Peru.
Okay. You are from Peru, members?
Yes.
Yep.
We're from Peru.
Okay.
Yes.
So again, you have 20 minutes. This is the presentation. This is a conversation with different judges. So any judge who wants to give a step forward? Maria Jose?
I can Okay, perfect. So nice to meet you, Peru. I really enjoy your work. And I'm just wondering, you designed a polycistronic circuit with one signal peptide and multiple RBS. So how could the movement of the transmembrane proteins can be affected by that? And how are you thinking to address this problem?
Oh, well, yes, as you said, we're using 2 cassettes, and in each of them has exactly the peptide production for 3 and the other for 2. At the beginning, we thought to use all of them and to put in 1 cassette. That's why we divided into 2. Then we modelized how are we gonna produce them. And based on that, we had the conclusion that, yes, it's gonna rise exactly the production, but not at the point that we are gonna reduce or have exactly like too many in the first peptide, and then the last peptide, it's not gonna be produced. It's exactly the perfect concentrations that we want to produce.
Yes, and regarding the single signal peptide, we did realize after we had turned in our work to the iGEM Design League, the problem here, and we have actually corrected it to put a signal peptide next to all of the peptides. Nadia, if you can share your screen, you can see the benchling, our benchling file already has that corrected.
Right. Here we have, and this is the final peptide that we corrected after we saw that problem.
Yeah, same with the his tags.
Yeah, this is our final circuit that we correct that mistake.
Amazing. So happy to hear that you were you realized that.
Mm-hmm.
Okay, that's it, Maria Jose. Okay, thank you very much. And next judge, please. Who wants to give a step forward? Alan, you go.
Yes, hi. Thank you for your presentation, for showing us your work. I was really glad to see it. I enjoyed especially how you interfaced with the farmers and people involved in the supply chain to understand this process of food spoilage. My question relates more to the application. How long do you expect your proteins to remain stable and effective on the produce?
I think I can answer that question. Since our product is going to be lyophilized, so it's its life will be really large, but this need to be tested in future lab experiments. But we expect like some years of duration of the product when it's lyophilized, and when applied to the fruits as a coating, we can calculate the half-life of the proteins during the antifungal activity assay. So we did a mathematical model, and with this curve we can actually measure what's the time in which our proteins are acting. So if maybe one of anyone else want to add something?
Yes, so for the stabilization of the lyophilized peptides, we would need to put special care into really making sure that our peptides aren't contaminated in any sort of way after they have been lyophilized, so that we can ensure this long lifespan of the product.
Perfect. Thank you.
Okay, thank you very much. Now let's go with William.
Thank you very much for your project and the wiki that you made for us. I just have one question. What fruits would benefit the most from your solution? And do you have any competitors within this space?
Okay, so for right now, we have focused on 3 fruits: strawberry, tomato, and aguimanto, which is a goldenberry fruit native to Peru. And we chose these fruits because they are the most acceptable to our 3 fungi that we have been focusing with our peptides. And regarding the competition, we have analyzed 3 main types of direct competitors with our project. It would be the chemical preservatives, it would be the freezing of the fresh products, and it would be the antimicrobial coatings that would be applied on top of the fruits. So Nadia, if you can share your screen to show the graph that we have made to compare these types of products. Especially, we took 2 criteria into consideration. It would be price, and it would be health benefits or health issues. So for the preservatives, these are considered very cheap. However, they can, in the long term, bring some health issues to the consumer. For the product freezing, this isn't an issue with health. However, it can be pretty expensive for certain companies to develop. And finally, the product coating has the same issues with the freezing. It can also be somewhat expensive. So what our product aims to do is be both a cheap option and also an option that doesn't inflict any health issues.
Sounds good. Thank you.
Okay, thank you very much. Another judge, please. Hector.
Yes. Hi, everyone. Congratulations for your work. What was your main challenge during this project, and why?
Can you listen to me? Yes. Okay, I think the main challenge was just to be here. We had many problems with the financial part, and we just do a lot of things to be here, like just selling products, selling food in our houses, and also just touching and just saying to other people that we need help to be here. So, and also to prove that we are able and we are capable of doing this. So yes, many people don't believe on us that we can do synthetic biology and a great project, but we are here to show that we can do it. So yeah, that's all. And also that we can obtain $1,500 by ourselves.
Great. Thank you.
Okay, thank you very much. Another judge? Felix, Mariana? Mariana, you go.
Hello, guys. I want to say that I really loved your project. I think this is a huge problem right now, because it's not a matter of not being enough food, but of how it is distributed. So I really enjoyed reading it. And I was wondering, right now, you don't know by how much will your product extend fruits and vegetables shelf life, right? So how would you assess this information? Did you hear me?
Yeah, someone from the MikuyTec team that may answer?
Oh, yes. Well, based to answer these questions, we already put on establishing protocols exactly to measure the antifungal activity of our peptides. Exactly, we are gonna know the exactly time that we can extend the food shelf life after doing those experiments.
Okay, thanks, guys.
Okay, thank you very much. Felix? You're on mute. Felix, you're on mute. Felix, you're on mute, so we cannot hear you. Felix, do you have any questions for the team?
Felix, you're muted.
If he has some problems with the audio, maybe he can write.
Yes, actually, I was writing him. So, well, actually, I just wrote to him, but I don't know. Felix, can you hear me? But do you have any question, or you're on mute, man? So. I don't know.
Maybe he has some difficulty. If in the meantime another judge, David has a question.
Yeah. Well, there was a. So I have a deck of questions. What do you think is the estimated price for the gold market? We're going to Maria Jose. Actually, I shouldn't make a question, but Maria Jose wants to make a question, so you go, Maria Jose. Thank you very much.
Okay, perfect. So I saw that in your first circuit you had the CBD, the chitin protein, in the C terminal. and then you change it to the N terminal. So how did you figure it out that it was a better idea to have it in the N terminal?
Can you repeat, please? I have a little lag on the Zoom audio.
I'm sorry. Is it not, okay, it's fine. So repeating the question, it's just that I saw the cheating protein, the CBD, you had that in the C terminal in the first circuit, and then you make some optimization and you put it on the N terminal. But I'm wondering, how did you make that selection? What happened to make sure that the N terminal was better?
Okay, we did some 3D modeling of the proteins. So we determine which combination of all the protein segments actually works in a better way. So that's how we choose not only the parts that contains our engineered protein, but also the order in which we need to put them in the sequence.
Listo, ya regresé. Tuve ahí unos problemas, unos inconvenientes.
Bueno, buenísimo. So, you have a question? ¿Una pregunta, Félix, para el grupo?
Ok, bueno, antes que nada me encanta su proyecto. Creo que de todos los que he visto es de los que más me ha gustado en cuanto a la prospección comercial que ustedes tienen. La verdad, muchas felicidades por ese trabajo que hicieron de investigación y sobre todo de encontrar cuáles son las posibles barreras a la entrada que tiene su producto, porque al final de cuentas, cuando hablamos de productos biotecnológicos, siempre vamos a encontrar que van a ser productos que van a sustituir a otros. Entonces, me encanta esta manera en la que ustedes lo han trabajado. Y en cuanto a los temas de divulgación científica, tal vez mi única ahí duda o algo que me hace un poco de conflicto es ¿cuál ha sido ese propósito de divulgación científica? Porque a mí me encantaría que todos esos talleres y las actividades que realizaron se pudieran hacer, pero enfocadas al mercado objetivo que ustedes quieren concientizar para que les compren. Sería lo único. Y mi pregunta radicaría en eso. ¿Qué tipo de estrategias hasta ahora de publicidad han tratado o encontraron para poder introducir su producto dentro del mercado que muy posiblemente les compre?
Yo puedo responder esa pregunta. Lo hicimos prácticamente porque en el país, Perú, existe un miedo constante a lo que son productos químicos. La población vive siempre atemorizada porque no es informada adecuadamente sobre los peligros o no es informada adecuadamente de qué es lo que le están poniendo a su fruta. Es justo cuando expusimos la infografía se nos acercaron muchas personas diciéndonos: están haciendo algo químico, nos van a dañar o van a perjudicar nuestro producto. Entonces, justamente la idea es generar confianza en la población, informarle qué es nuestro producto, educar y luego para luego transformar, ¿no? Entonces, de esa forma introducimos un producto no solo de forma de un producto, sino un producto con un concepto de educación también, o sea, hacia la población, hacia la aceptación que va a tener hacia nuestro producto, ¿no? Más que todo porque las personas que nos entrevistaban o hablaban con nosotros nos decían, ¿no? Tenemos miedo, no sabemos qué le están poniendo nuestra fruta, nuestro producto. Entonces, justamente la divulgación sirve para eso, ¿no? Para democratizar la ciencia y hacer de que pueda llegar a todo público y se pierda este miedo generalizado de lo que es, por ejemplo, el SynBio.
Do you want me to translate?
Yes,
please.
Yes, please.
Okay, so in summary, what we want to do is to change the fear of most people that they have towards biotechnology. What we did is to do some presentations, and the people that came to us, they were asking, is this chemical, is this going to hurt us? So our goal with this is to actually make people to understand the science behind it first, so that they will buy our product. And second, which we consider is the most important part, is so that we can democratize this knowledge and we can change the world in a certain way. So that is something that is not just for a special group of people.
I would like to add something to that, and let's understand a little bit the context of Peru, because we are usually restrained by the law, because in Peru it's not legal actually to produce something genetically modified alive and do a business with it. So this is one of the reasons for us it's so important to democratize also the synthetic biology and biotechnology itself. so more people can know what is about this knowledge area, and they lose this fear they have, and also to try to ban this law that is currently established by the government.
Ok, gracias. Felicidades. Ustedes primero están considerando es el usuario, a final de cuentas, no es a quien le venden, pero es el usuario final y a quien deben tratar de convencer. Me agrada su proyecto. Muchas felicidades.
Well, thank you very much, Felix, and thank you very much to the team of MikuyTec. First time, I guess, Peru in iGEM, right? Or second? Very first time. Well, congratulations. You made a great job, and hope to see you next year in Paris. So right now,
thank you.
Thank you.
Thank you.
Nice to have you here.
Thank
Great
you.
job.
Thank you very much.
Thank you.
So right
Thank
now
you.
we're gonna have a break of 5 minutes for the judges, and then we come back with the Ollin SynBio IPN team. So judges have 5 minutes for go and do whatever you want, and in 5 minutes we're back, right? I'm gonna ask gently the team of MikuyTec to leave the session, and thank you very much.
Equipo Ollin, Paola Galán es de su equipo.
No.
Ahora sigue el equipo Ollin SynBio. Estamos en un break de 5 minutos y volvemos. Por favor, a los que acaban de llegar, pongan su nombre, renómbrense ustedes mismos poniendo el nombre del equipo y después el nombre personal. And if you're a PI or an instructor, please add to your name that you are a PI or an instructor, please. Well, this is a call for all judges. Please come back to the session. We're gonna start right now with the team of Ollin, SynBio, IPN. Please, only members of the IPN team and the rest of the last team of members, please leave the session.
You ready?
I'm just gonna call each judge to not be here. Alan.
Hello.
Hector.
Yes, I'm here.
Mariana. Mariana, are you here?
Yes, I'm here.
Okay.
I raised my hand, but I guess you didn't see it.
Okay, thanks. William? Emiliano? María José? Will María José, are you here? María José Durán? And I don't know. In Felix?
Aquí.
Okay, perfect. Okay, so we're Ollin, SynBio, IPN. You have 20 minutes. This is not a presentation, it's a conversation that you're gonna have with the rest of the judges. Please try to keep it short, your answers, so we're gonna have more time for judges to make more questions, sorry. So I don't know, any judge who wants to give a step forward? Alan, you go.
Thanks. Hi. I really liked your project. I think this is really getting at the root of accessibility in synthetic biology. And you really, it shows you took the time to really think about how to not only lower the cost, but try and make it super easy for teachers and educational institutions to access this much more easily. So congratulations for this. My question then relates maybe more to the final implementation of your project. So let's say toward the end of your presentation, right, you have an image of you sending these distribution boxes out, etc. Pretend I am a teacher at a high school or a college, and I want to show my students how to do a cell transformation. Can you tell me briefly what I would do and what would the process look like? How would I potentially order this? How do I run the protocols, etc., to have a complete project for my students?
Yes, well, the ordering of the boxes will, we're planning to launch a website in the future where we will put the boxes up for sale. So the user or client can buy the box and then it can get delivered to the user. Maybe someone from HP or Wet Lab can implement, well, can comment how the implementation would be done.
Yes, so we want that the same institutions are capable of producing their own reagents. So we are actually providing them with the vectors and the biobricks for the reagent production. So it will be the institution that produces its own reagents that then could be used for practices for students in high school or in university level.
So reagents like growth medium, broth, things like that, right? Okay. Yeah, thank you.
Thank you, Alan. Now let's go with Emiliano.
Hi. I wanted to ask, just out of curiosity, why Cas9 or other CRISPR enzymes are not listed?
at the beginning, we did consider including CRISPR or Cas9, but as we as students in university level didn't have the chance to work with them, they were difficult for us even to understand. So I was like, okay, how are we including something that we really haven't had the time to work with? So we decided to focus on what we knew, and then we were like, okay, we have work with cell transformation, with PCR. So that's what we were including, what we knew we could work with, and later on we can include more things.
Okay, yeah, I was afraid there was another regulatory issue about the patents of this system, but thank you.
Thank you very much. Now let's go with Mariana.
Hello, guys. I really liked your project and your approach to the accessibility of synthetic biology and biotechnology in general to younger audiences. But I was wondering, I saw that you decided to re-engineer the Taq polymerase. Why was that? Like, I assume, well, I can read here that you decided this to achieve better fidelity and better results, but why wouldn't you use another polymerase?
Well, why Taq? Well, Taq is the most used polymerase for PCR reactions, so we decided to work with it, and then we decided to do the re-engineering of it because we had meetings with the possible stakeholders, and they told us that they wanted polymerases for retrotranscription. So we were, okay, should we include another enzyme? And then we searched it, and it was actually a little bit difficult to produce retrotranscriptases. So we decided to modify Taq in order that it can either work as DNA or on RNA, because we found these articles that made these punctual changes in Taq that made it possible for it to work as a retrotranscriptase. So, and we also made other changes to make our Taq more stable and more efficient. So we want that this, let's say, ambivalent protein that can even work for PCR or for RT-PCR.
Okay, I get it now. Thank you.
Okay, thank you, Mariana. Now let's go with Maria Jose.
Yes, Mariana, I had the same question that you had, just because I think that the whole process of modifying that enzyme can be like expensive and like put you in a situation. But anyways, I had another question for you, and it's about your vision. I totally agree with you about the importance that it can have, that this package can have in helping other countries besides Mexico. But talking about that, what do you think about the legal permits that you have to consider? How are you gonna make to put your product in a better position than big companies that already have like the legal permits solved?
We'll ask for, I mean, I guess you're referring to exportation. We would only be exporting DNA, plus DNA. So we don't see there would be an issue regarding that part. Someone from HP can complement this question.
Yeah, sure. Yeah, actually, we consider at one moment to also provide the chassis for the transformation, that being Escherichia coli. But we decide against this because that imposes more barriers, or that makes more difficult the distribution of the box to the students. And so there being only plasmids, there only being DNA in the boxes, makes it a lot easier to make them accessible to the students, because in the Mexican regulation, it is considered as a chemical reagent. DNA is considered actually as an acid rather than that being a biological reagent, and that let us make it more accessible to the students, as I was saying.
Okay, thank you very much. Now let's go with William.
Thank you very much for your project. I just wanted to find out what is the cost relative to other any other competing products.
I'm sorry, I had some lagging. Could you please repeat the question?
No problem. What is the cost relative to competing products, and are there any competitors?
Yes, our expected cost would be $250 per box, and comparing with Merck, New England Biolabs, and Thermo Fisher, we are well, we made an analysis, and that would cost around $700, the minimum. So this would lower the cost by a lot, and well, we're more thinking of us like as an alternative more than as a competition. Given that we're aiming towards a different purpose, but yes.
Sounds good. Thank you.
Thank you very much. Now let's go with Hector.
Hey, hi everyone, and congratulations to the entire team. You are doing a great job of inclusion and access to synthetic biology. We know that access to reagents and laboratory equipment, especially molecular biology, are very expensive and take a long time to be imported. I would like to know what is the main limitation of your project and why?
Well, this year limitation, for example, for starting our project, we had two limitations. It was the COVID restrictions and finances. So our university didn't have much of a budget for us to start a project, so we actually had to go crowdfunding, and our initial goal was to enter to regular iGEM, but we didn't achieve that. Well, the price, it was too high, but then there was this opportunity for iGEM Design League. And the second was COVID restrictions. So the university is closed, so we didn't have access to laboratories. So we relied all in silico essays, and we worked hard on bioinformatics for our project.
Okay, and suppose that money is not a problem. What will limit you to carry it on?
Okay, I don't know if the question refer more to the limitations of the project to implement it in schools. Was the approach you wanted to ask of, or?
I don't know, maybe technical limitations?
Okay, well, the infrastructure of the schools is still one difficult challenge because a lot of schools in Mexico don't have the equipment to make this kind of experiments. We are working on that aspect too. We wanted to focus on the reagent side of the problem rather than the equipment side this time, to focus our energies first solving that part, but we are working with people in Mexico that have developed low-cost equipment for using at schools for this type of experiments, and I think that is one side of the problem that we are probably in the future going to work on. But that is one of the limitations that still exists that our project doesn't fully cover.
Thank you.
I don't know if Sinue wants to add something. Yeah.
Yes, in addition to what Eduardo said, we identified this through the interaction with the stakeholders. The stakeholders was the people that lead us to notice that there are some limitations to implement the Tettle box in schools of the bachelor's degree. So we identified this, and we are actually working in collaborations to try to make this not a problem, because there are some other groups that are working in like micro labs, and we are now chatting or talking with these kind of teams to collaborate and make this problem less, a little bit not too difficult for us.
Okay, thank you for your answer.
Thank you very much. Felix, you have any question?
Sí, gracias. Bueno, antes que nada, felicidades por el proyecto. Me encanta toda la versatilidad que de hecho creo que puede tener su proyecto. Justamente basándome un poco en las preguntas de mis demás compañeras, especialmente de Mariana, que me encanta, estoy enamorado de Mariana, debo admitirlo. Pero, bueno, aquí el punto es, ¿ustedes qué han encontrado que sea su ventaja competitiva? O sea, más allá del costo y más allá de ciertos beneficios, ¿cómo ustedes pueden identificar esta ventaja competitiva respecto a las demás marcas que ofrezcan algo parecido a ustedes, y especialmente a marcas que vengan, que sean parecidas a ustedes y vengan a competir de la misma manera en precio y también en la calidad y en los, por decirlo, experimentos o actividades que tiene su producto?
Well, first of all, the unique value proposition of our product is that we're letting the user create their own reagents, which would lower costs drastically. This will also have a lower shelf life of reagents, meaning that, well, they have their bacterial cultures, their bacterial transport cultures, and they can store those cultures and they can use it for the posterity. However, if you buy from traditional or reagent producers, well, you use it for 1 or 2 practices and then it's done. You have to buy more, and with this, you have it stored and you can create more with the transport cultures.
Ok, súper. ¿Y han encontrado ustedes también más que un mercado objetivo, más bien momentos de consumo para su producto? Bueno, hoy en día parte justamente del marketing holístico nos dice que más allá de vendérselo a un grupo de personas o a personas con ciertas características, necesitamos momentos específicos de consumo de este producto. Esto para poder diversificar y expandir las oportunidades de ingresos para una organización. Entonces han logrado identificar momentos en los que su producto pueda ser consumido y no sé, alguna variante, por ejemplo, que ustedes mismos puedan generar talleres o cursos o puedan crear este tipo de laboratorios móviles a manera como una empresa STEM, ya más que solo de la venta de los kits.
Sí, en efecto, se han encontrado instituciones claves en las que comenzaremos con la difusión de estas cajas. Lalo. Wait.
I'm going to translate for Daniela. We found some key institutions that want to work with us with the utilization and the distribution of the boxes.
Nuestra caja ha sido evaluada por otras personas y aprobada también. Con esto conseguimos cartas de intención.
The idea of the boxes have been evaluated by other people from these institutions, and they have provided us with intention letters.
Y para esto en una de ellas se hizo el convenio de tener ciertas cajas para estados del país de México y a su vez continuar con la difusión en otros lados, teniendo alianzas para su posterior donación.
In these intention letters, we came to an accord with the institutions to provide a certain number of boxes to a certain region in Mexico, and then continue working in this type of relations that would benefit both the institutions and us in making the boxes more widely available.
Además, pues en 5 años pensamos que estas cajas ya estén en el mercado.
We expect that in 5 years, or that is the business plans we lay out, that the boxes would be part of the market.
Ok, 5 años, o sea, este proyecto está prospectado para dentro de 5 años ya empezar a vender.
I'm sorry, I'm going to cut it in this because we have another question. So in order to give more time to some other judges. So, and we are almost over time. So Mariana, I'm sorry, Felix, and let's go with Mariana with that. She raised her hand many minutes ago, and keep it short, please. And we finish with you.
Yeah, real quick question, guys. I was thrilled and so happy to read that you were proactive in the creation of a bioethics committee at your uni. So I was wondering, how is this project going?
Well, this project is going slow but sure. We encountered many roadblocks while doing this project because it's not as easy as we thought. But we're still working with teachers and our school's principal and the school's council to keep developing this, because we did the research to know the steps we had to take in order to complete the project. But there are many other aspects, internal aspects from the school that we need to learn. So we are working with our teachers and the principal and the next generation of our team, and the students that continue in the team will continue this project, and we are hoping that by next year the committee will be implemented.
Great, congratulations, guys.
Well, thank you very much, and thanks to Ollin, SynBio, IPA team. It was a pleasure to have you here. And now we're moving to the last but not least, the Panama University. Let's give a hand to the team, and let's go with the next team with Panama University. So please, Panama members, remember to rename yourselves as the name of the team, then your name, and if you're a PI or instructor, please add your name in order to know your level, please. And to the members of Ollin, SynBio, it was a pleasure to have you here. And I'm going to ask you gently to leave the session in order to leave space to other people.
Hello again, judges. Just as a reminder, as soon as the next session finishes, you will have some time to discuss your thoughts with the other judges. I imagine you have a lot of ideas right now going on, so you will have 15 minutes, 20 minutes, half an hour, the time you need to discuss the session with the rest of the team, of your judging team. So, David, please just, when the next team is done, just finish the live stream and also the recording, and you're going to start the discussion freely.
And in secret.
In secret.
Okay, thanks. Thank you. So I don't know if the team of Panama is ready. Remember, please rename yourself with the name of the team and your personal name. And if you're a PI or instructor, please put to your next to your name exactly edited. I'm seeing you. put PI next to your name, and we're gonna start in just a couple of minutes. Well, we're gonna start with the last theme of this first class of iGEM design. It's a pleasure to have you here. Thanks to the judges for all the time that you have give us today. We have back this team of Panama, and well, we have 20 minutes. This is not a presentation. This is a conversation with the judges. So I don't know if any judge wants to give a step forward. Maria Jose, you go.
Yeah, so nice to meet you, Panama. It was wonderful to read all your work. Congratulations for that. Something that got my attention is that you're using 3 different proteases and also 3 different extraction columns. So have you considered using just one protease in one sense, and in the other, have you considered other extraction methods with other physical or chemical properties that can substitute the other 3 ones that you have?
Yes, so basically we use these 3 different proteases just because we want to try each one of them. So they're basically like going to be used for tests when we do, if we get this project to lab environment. So we're gonna see which one is the best, like which one is the one that works best, that has the better conditions to produce these cuts. And what was the second question, sorry?
The extraction method, because you also have at the end 3 different tags.
Can you repeat it, sorry?
Yes, sure. So at the end, you have 3 different tags to extract by 3 different methods. So have you considered other extraction method that can separate all of them by chemical or physical properties?
Yeah, I think it's the same concept. We want to test them all out and see which the best one. Obviously, we could use other extraction methods like HPLC and stuff, but we haven't really checked these other methods. So we're just trying to test everything out. And that's why we have so many different parts, because we want to test each one of them and see how they work. That's part of our experimental model. So what we would like to do if we get this to a laboratory.
Okay, thanks.
Okay, next judge. Someone who wants to give a step forward.
Hi, I just have one question. In what are you testing your product once you have it? Because if it fails, you will ruin maybe some important document from Simon Bolivar.
Yeah, of course. We're not gonna go straight away to important documents, because that would be irresponsible. But what we will try to do is to test it in regular paper with cuts that are similar to the ones that we see when there's degradation. So, for example, like little holes in the paper, we can make them with a punch, like a something that punches the paper, and we make like little holes. This way we can avoid do this in documents while we validate our experimental model.
Or you could inoculate conidia from a fungi. Okay, you answered my question. Thank you.
Yeah, sorry. Just to clarify, we will make antagonism tests to test like, okay, so this antifungal protein works inhibiting this conidia stuff. So yeah, we already have that part like sorted out. Yeah.
Thank you.
Okay, thank you very much. Next judge, please. Someone who wants to give us the forward?
I can ask a question. Thank you very much for your project. I just wanted to find out, what does the lacase enzyme do with respect to the degraded paper? Is it related, or is it just for producing new paper?
So the thing that we have talked with the librarians is that we're talking about the lacase, right? The lacase protein. Yes. So when we talked to librarians, they told us that lignin is a part of some paper procedures that makes paper brittle over time. So when they buy paper, what they do is they try to buy lignin-free paper. So what we're doing now with the lacase, this lacase will degrade the lignin into subproducts that are soluble in water, I think. And what we will do is we will be able to wash off the lignin once they're treated with this lacase. Alan has a question.
Yes, I think Mariana had her hand up first.
Yeah, let's go with Mariana, and then we go back to you.
Okay, great. Well, guys, I was really happy to read through your project, and I think it's a perfect example of how SynBio can help at various areas of opportunity. So I was really happy, and I thought it was really original. But I was wondering if your final product could be applied to paper before it needed conservation, like in a way to prevent this kind of degradation, and not once the conservation work is needed.
Yeah, so yeah, we've thought about this. We have thought about making maybe like packaging, biosustainable packaging that could be used to resist degradation of this of fungi, for example. And yes, of course, I think that would be a great idea, and we should test it first before we implement it. So yeah, of course, that would we can dream of things like this, but we first have to validate it. But yeah, of course, it will be great.
Okay, guys, good luck.
Thank you very much, Mariana. Then we go with Alan.
Yeah, so thank you again. I really enjoyed looking through your project. I have maybe a bit of a naive question, but you hinted at this, that this is not just unique to Panama, right? There's old documents everywhere in different conditions and different states of preservation. How robust do you think your system is to these different types of paper, right? There's different lignin contents, different hemicellulose contents, et cetera, there's papyrus, things like that. Do you have an idea of how and if you need to adapt to different types of paper?
Yeah, so we're focusing on this project in cellulosic papers. So we're just working with papers that come from wood or other cellulosic materials, and just working with that. Of course, there are different types of papers that contain important information, like as you said, papyrus, but they come from different processes. So this will be a little bit more difficult. Either way, when we talk to the curators and the people who know more of the matter, they told us a little bit about different papers that are not that common, and sometimes they do use the thief caster to restore this kind of papers. But it would have to be, again, validated. Everything has to be validated because we're dealing with something that's very precious and invaluable. So we would rather first test it and then go progressively towards other different types of paper or document containing sheets. Yeah.
Thank you.
Thank you very much. Now, Hector.
Hi, everyone, and congratulations, Panama. You're doing something very good, very great, and it is a problem that I never imagined that synthetic biology could solve. I think it is a great success. I only have one question regarding to entrepreneurship part. Is the market large enough to make an investment in research, development, and production of your product?
Well, his name is, you said right now, but the guy that was answering just now is Daniel. Yeah, I'm sorry.
Sorry.
Well, so everything we have thought of, it's in relation to what the curators and restoration professionals have told us. So for that, we have made this very special, tailored kind of process. What we have thought of is to first give them like some validated prototypes and for them to try them and keep exploring, not only here in Panama, but our entrepreneur business model was based on like what we've been talking, the people we've been talking to, and the librarians and other individuals that do this kind of curation and restoration, like art restoration, for example. And yes, we think it could be large enough for us to apply them. We just need to reach those restorers and librarians.
Okay. Sorry, I also want to add that again, and sorry to like punch this in a lot, but we have to validate it all. So if we can produce or we can make a high enough quality product, we definitely are gonna reach a bigger market.
Thank you.
Thank you very much. Now, Felix.
Felix, you're muted.
Felix, you're on mute. Felix, can you hear? We can't hear you. I'm gonna write him down. Felix, can you hear us?
Listo. ¿Ahí se escucha?
Listo, buenísimo.
Ok, súper. Bueno, antes que nada, bueno, como decía, felicidades.
Felix, we have some echo coming from your video.
You maybe have devices, so you have to turn off one of them.
¿Ahí ya se escucha mejor?
Ahora todo bien. Ya puede preguntar, listo.
Ahí ya se escucha, ¿verdad?
¿Perdón?
¿Ya ahí se escucha bien?
Se escucha el eco, todo, ya se escucha bien.
Ok, perfecto. Vale, bueno, como estaba diciendo, antes que nada, felicidades a Panamá. Me encanta su proyecto, me encanta que sea tan específico, es bastante creativo. Entonces de ahí tengo solamente dos preguntas. Una es saber de dónde viene este interés en la restauración y en su tema principal del proyecto. Cuando hablamos de algo, de un proyecto, de algo que va a un mercado tan específico, yo siempre le digo a mis clientes que es muy importante trabajar en la esencia de la marca. Entonces, es importante saber cuál es la motivación, porque eso nos permite conectar mucho mejor con este público que es tan selecto. Entonces, quiero saber de dónde viene esta motivación por este tema de la restauración.
Should I answer in Spanish or?
We have time actually to give in Spanish and in translation, please.
Ok, bueno, pues inicialmente nosotros habíamos pensado en hacer una clase como de tape que pudiera solucionar problemas rápidos sin tener ese como molesto brillito y así. Después seguimos explorando la idea de que este tape como que se fusionara con el papel, y ahí fue cuando decidimos visitar a la biblioteca de la universidad de la mitad de nuestros team members, y allí fue cuando nos dimos cuenta que el problema no era ponerle un tape y ya, y listo, sino que no solamente ellos tenían problemas para restaurar esos huequitos, sino también para conservarlos. Entonces tenemos que darle una respuesta más integral a todo lo que ellos tenían, pues, porque de eso trabajan, de eso comen. Y nosotros, pues, de alguna u otra manera, todos estamos agradecidos. Paréntesis, en Panamá es el mes de la patria, porque todas nuestras cositas caen, todos nuestros eventos patrióticos caen en un mes. Entonces, todos de alguna manera estamos agradecidos porque ellos siguen ese hilo de conservación, y no solamente eso, nos vimos mucho más inspirados a seguir con esto, porque todas las personas eran adultos, digamos como mayores de 60, 45 más o menos, y muchos se quejaban de que no había nadie ya que fuera y los ayudara con su problema. Ni siquiera el gobierno tenía como interés en hacer esto, entonces nosotros vimos una manera como más personal de poder solucionar su problema y asimismo llevarle ese como relevo generacional, incluyendo partes de biociencias. Y pues mucho más, porque también necesitamos un hardware para aplicarlo y otros.
Para añadir a la solución de este problema, pasa a un ámbito mucho más grave, ya que el problema tiene una afección para su salud, ya que estar en contacto con este tipo de hongos patógenos, ellos podían verse afectados teniendo algún síntoma de asma, algunos nos habían comentado que podrían tener incluso infecciones en el oído como otitis, y que verdaderamente esto suponía una afección mucho más allá de solamente el problema con el papel, sino directamente con el problema de salud con ellos mismos.
Ok, súper. Me encanta su idea y cómo conectan con este proyecto. La otra pregunta es si han encontrado o tienen un plan, por ejemplo, una ruta de cómo llega su consumidor a comprar estos productos, estos servicios. ¿Cómo se acercarían ustedes o cómo normalmente los productos que ya existen para conservación llegan a este consumidor? Si ya tienen ese plan y tienen alguna idea diferente de cómo abordarlo.
Bueno, primeramente después de validar, disculpen, nuestro prototipo, se lo daríamos a pequeños, como a individuos que se dedican a estos individuos profesionales tipo curadores de arte, y así luego más tarde, después como de probar varias como ideas que les suplan sus necesidades, estaríamos targeting las bibliotecas y otros tipos de espacios de restauración para llevarles como productos más masivamente, que estamos seguros que funcionen y pues que a ellos también se les haga fácil su aplicación.
También quería agregar, perdón, que también sería, pues, o sea, parte de de lo que ya hemos estado haciendo, de conversar con los restauradores y esto, ya tenemos como contacto directo y tenemos relaciones más cercanas con estos curadores, que sabemos cuáles son sus necesidades. Entonces, en cuanto ya sepamos cuáles son las necesidades, bueno, como ya las sabemos, podemos suplirle esa misma necesidad. E igualmente, como dijo, a medida que vayamos validando, vamos haciendo un producto que es cada vez más robusto y por lo tanto vamos llegando hacia el mercado que ellos necesitan. Y obviamente se va a expandir a medida que tengamos como esta alternativa, ¿saben? Entonces le damos la alternativa y ellos van hablando en el mercado porque igualmente ellos siempre tienen como reuniones o talleres que son internacionales, entonces se va hablando, hey, mira, nosotros tenemos esta alternativa que se hace, es menos costoso que comprar papel de China, papel china se llama, perdón, papel japonés, disculpen, perdón, me equivoqué. Entonces, este papel japonés es súper costoso, entonces tenemos esta alternativa que es más barata y se produce en Latinoamérica, y todo esto entonces nos puede ayudar a llegar al mercado mucho más fácilmente y rápidamente.
Okay. Excelente, muchas gracias, Panama.
Thank you very much, Felix. Now, Maria Jose has another question.
Yes, thanks. So some antifungals in high concentrations can lead to PKS growth affectations, right? So what are the main risks that you expect to see in vitro? And then what are your plans to maximize the protein production?
Sorry, can you repeat the question, please?
Yes. So some antifungals in high concentrations can lead to PKS growth affectations. So what are the main risks that you expect to see in vitro, and what are your plans to maximize that protein production?
Yeah, so we have already thought of ways to see protein production. So for example, we have different types of, well, different strength promoters, especially constitutive promoters and different strength RBSs. So as we start to make cytotoxicity assays, we will start seeing what's the concentration at which our PKI will start to have growth problems. And so we have to obviously find a balance in which we can produce enough protein to make to have the effect we are looking for, but also that does not kill our PKI. We also tried to use the SEGI protein. So this protein will only affect specifically the cellulosome mechanism. So PK does not have this mechanism. And this way we can avoid the death of PK. But there's the other antifungal, which is really long key smash. So this one, we're going to have like more precautions with it. And I think it will not, from literature, it will not affect that much PK because it affected some other fungi. But of course, it's something that has to be tested. We do not have the real answer because it has not been tested against PK. So it will be just a matter of testing and seeing the concentrations. Yeah.
Amazing. Thank you.
Thank you.
Thank you, Maria Jose. And we still have time for one more question. I don't know if any judge wants to give a step forward. One last question.
Have you thought of another applications aside from librarians? For example, for the fungi that are in my wall in my bedroom, like a spray?
Yeah, sure. We have thought also about wallpaper. So wallpaper has a different type of, let's say, composition. But for some types of wallpaper, we could use these antifungals. Of course, these are broad action antifungals. I don't know if that's the correct answer of the word, but they act in a high, in a different variety of fungi. So it shouldn't be a problem to apply it, but we also have to take into account that one of our antifungal proteins will only affect fungi that use the specific cellulosome mechanism. So yeah, of course we can apply to anything, even medicine if we could want to, or like agriculture. But yeah, that's basically it. We can test. That's what science is for. Test everything. No puede.
Okay. Okay. Now we really have time for one very last question to close this session, this first class of iGEM design. So I don't know if there's any judge who wants to ask something else. And with this, we close this session.
Guys, I have one question. Maybe you can answer it quickly. I saw that you decided to use SDS page as a confirmation mechanism. Why did you choose this confirmation mechanism, and can you think of any others?
I'm sorry, what mechanism?
SDS page.
Yeah, for what?
For your protein production.
Yeah, it's easier for us. It's the standard, I think, procedure that we can think of. Of course, we also have the fluorescent reporters, and we can also use this as a way of seeing or quantifying our proteins. But yeah, of course, we could. Yeah, it's just because it's the simplest one, I think.
Okay, thank you.
Well, thank you very much. Thank you very much to the team of Panama University. It was a pleasure to have you back here in iGEM Design. And with this, we close this first class of iGEM Design. So I think we all deserve a single applause.
Oh, hey! Woo!
Congratulations, Team Panama.
For all the time that you have given us today. And I'm going to ask the judges to stay for small conversation. And thank you to the Panama University. We're going to ask you gently to leave this session.
Sorry, can I add something?
Please.
I think that no one talk about the biosafety and biopolicy work that you did. It was amazing. And I wasn't expecting less than that because you have like your great mentor there, but it was impressive. So just congratulations because of that.
Thank you. Thank you. He's the best. Thank you, everyone.
Have a nice day. I will end the stream now.